Let me start the record button. First, thank you very much to Professor Jessica Opie, who heads the Hematology Laboratory and Hematology Division in the NHLS at UCT. She's my co-collaborator, conspirator, partner in crime with everything we do in Cape Town. And without her and her team, hematology would not exist at UCT. So Jessica, I want to welcome you both as a colleague and a friend. It's really great that you can be leading the meeting today. And I'm handing over to you to introduce our speakers, and Jessica will lead the conversation as well. Thank you.
Thank you very much, Prof. Lohr. It's really an honor to be chairing this session. We're going to be speaking about molecular hematology today, and the first speaker is Karen Shires, who I've been privileged to work with for 15 years. In fact, both Theo and Karen I started with in 2005 in the NHLS, so we go back a long way. I think Karen is very well known as an excellent teacher, leader of our molecular diagnostics lab. And she's going to be taking us through some very, very practical day-to-day, I hope, interpretation of diagnostic reports. So Karen, over to you, making sense of the nonsense. Thank you.
Hi, first of all, I'd like to say thank you very much for giving me this chance. It's a pity that we're starting off molecular on this note, though. Instead of kind of advertising and telling you how wonderful molecular can be and how brilliant it is in a hematology pathology environment, I'm actually giving you a talk on all the pitfalls and how you have to be careful with when you request molecular. But never mind, hopefully I'll get an opportunity some other time to boast about all the wonderful things we could do. So I'm just going to share my screen, hopefully. Okay. Sorry, now I can't find it. Okay, can everybody see that? Somebody give me a yes. Hello?
Perfect, thank you.
Perfect, thank you. Okay. So today the title of my talk is making sense of the nonsense, and it's really more of a touch rather than anything, based on the fact that I often get asked by people kind of around the country just for some help on trying to interpret molecular results. And I've realized that there really is a basic lack of understanding because things are moving so fast. So hopefully this will give you a couple of slides just to kind of point out what you should look for and what you as clinicians and pathologists must start to take responsibility for, or help us out anyway. So things are moving really fast. We've got mutations helping with diagnosis. We've got mutations for risk stratification, therapy now linked to specific mutations, and now, which is fantastic, we're able to monitor responses. But this obviously comes at a cost in basically everybody's having to learn on the job, including me. The advantage of myself is I don't have to care about the patients as well, so I can concentrate on giving advice and getting the best kind of knowledge. I can accumulate the knowledge to actually be able to interpret and this molecular data. But obviously this is becoming more and more tricky for clinicians and pathologists. So as I say, things are getting complicated. So from what to test, should you be, what to test, when to test, should you be testing diagnosis, for instance, after induction, during maintenance, that's getting now confusing. What samples to use, blood or bone marrow, people like me continuously making your life hard, asking for things on ice or within 3 days. And how to test, whether you should be using karyotyping, is that good enough, microarray, NGS. And I think Theo will also point out later, that the number of testing platforms is expanding exponentially. And while that's fantastic from a research point of view and our capabilities, it is getting more and more confusing at the actual clinic level. So I actually put together this molecular hematology online course. I'm putting a bit of an advert here, which hopefully kind of has been adding topics on what to test, when to test, and how to test. But then I realized that obviously how to interpret the results, this is now, which seems kind of obvious, isn't. And I'll now explain why things are not as easy as they appear. So I think the biggest problem that we have is that, sorry, I just need a screen pointer. I think the biggest problem that we have, and I think one that kind of especially in the pathology setting is very unusual, is that molecular hematology tests are not standardized. It's not like an FBC or something like that where you have these gold standard automated tests. Molecular hematology tests are not standardized. Even good old BCR-ABL quantification, although that is, we've still now currently got different platforms for that. So that is our biggest problem in the lab. And because of that, most of us don't use the same methods. And because of that, the sample requirements are not the same and the assay sensitivity is the same. So asking for a test in one lab isn't necessarily going to give you the same result necessarily, or they're asking for the same samples as another lab. Unfortunately, and I'm really not picking on anybody, and I'm not saying we're perfect, but not all molecular hematology labs are equal. There has been an explosion in the number of kits and some equipment that is definitely making it easier for people that are not trained in molecular biology to be able to do these tests. But definitely there is a problem then when it comes to the expertise and the knowledge for interpreting the data. Very much it's rubbish in, rubbish out. So, you know, you really do have to have a fundamental idea of how the tests work. And I have used a lot of these kits in the past and often thrown them out the window because they actually don't do what they advertise, which is typical. Very much, there are lots of kits very much like the very mark, you know, magic mark type of scenario. So not all labs are accredited. As I say, we all have difference in reporting style, although there are recommendations. And I think the biggest thing is this, we don't all have the same level of expertise, and this can cause a problem. I think one of the bigger problems, though, is that clinicians don't give the lab any or limited information, and this makes result interpretation by the lab very difficult. While some things are easy, positive or negative answer, other things are actually quite difficult. And you actually, especially when it comes to quantification assays, and you really do need to have a little bit of information to be able to interpret these results properly. So I think the responsibilities of the requester, which I've never really spoken about before, but it's becoming more and more important. You need to understand what you are asking for and why. So that would kind of thing, like if you want to do a mutation analysis for diagnosis, then you need a qualitative answer, for instance. So you can choose a lab where they do a qualitative assay and not a quantitative assay, which tends to be more expensive. You know, what mutations do you actually need to look for? You know, just because you can look for something doesn't mean you should if you don't have any option to be able to change therapy, for instance, then that's actually, there's no point in doing it. But you need to know what mutations you look at, what you want, so that you specifically ask for them. And whether or not you are looking for minimal residual disease, for instance. So you need to know what level of kind of detection you're interested in. Choose the best job for the lab. The best lab for the job. So this would be, you know, I'm all for accreditation. It's a pain in the neck. It costs a lot of money. I sometimes hate it. We have an internal audit this week and I hate it, but it really does. It's really important, in other words, to make sure that you have the appropriate expertise, that the lab is basically having is peer reviewed using international external assessment, and that's getting more and more important when it comes to accuracy of quantification and mutation detection. Choose labs that give clear reports, that give you know, descriptions of methods and assay limitations. Those are really important. And choose labs that take sample type and integrity seriously. I'm saying this because unfortunately there are labs that don't, and that concerns me. And it really is up to the clinician or pathologist to kind of make sure that they're choosing the right people for the right job. And give the lab give the lab some information. Quick note on the request form: what disease is suspected? Is the sample diagnostic? This is a huge one. And at what point in therapy is the patient? That all helps in kind of trying to interpret the results. Sorry. So when it comes to now qualitative assay interpretation, so we really basically have two different kind of major types of molecular assays: qualitative and quantitative. So when it comes to a qualitative assay, this is basically where you're going to get an answer back that's where it says detected or not detected. And I think the important thing here is that people realize that qualitative doesn't necessarily mean that the assay is insensitive. And definitely I've had questions like that before. As you can see from this, where these are the different methods that could actually be used to give you a qualitative yes/no answer. They're very varied, even from NGS, even to nested RT-PCR, which is incredibly sensitive. These can actually all be used for just a yes/no answer. So some of these, as I said, are incredibly sensitive, 5-6 logs. That's a 6-log kind of sensitivity range. But what's important here is that different assay types can give you different answers, and depending on the assay sensitivity, even for a qualitative assessment. And this is where it's really important to kind of know what test you're asking for and to understand on the report when it says a limitation of an assay when something is not detected, what does that mean? So this is just a quick example. Say, for instance, somebody is asking for a P210 assay, and you get a report back where you have absolutely no information on the method that was used. So you've sent in the patient, which you suspect has got low-level P210 disease, so say for instance 0.004%. If you send it to lab A, where they do a FISH, they're not going to detect it. The sensitivity is not enough. Lab B, who use a multiplex reverse transcription PCR, that has a sensitivity of 0.01. That's also going to come back negative. But if you send it to lab C, where they use a nested reverse transcription PCR, you're going to get a positive result. So it's really important that on the report that you know what the sensitivity was. And if you don't get that on the report, ask. That is really important to be able to interpret these results. So when it comes to qualitative assay interpretation, they really cannot be used for monitoring. They're either not sensitive enough, even though I showed you just now that some are very sensitive, but for those that have like a 2% to 10% sensitivity, these are not sensitive for minimal residual disease, where you need to look for log changes. Or the problem is where an assay is super sensitive, like a nested real-time PCR, for instance, it's going to be positive over too large a range. So you actually have no idea where you are. So that's not useful for monitoring. As I said, it's a yes/no answer only. And be warned, and I had to put this thing about the PAX gene based on last week's talk. If labs are willing to accept samples after 3 days for analysis in really any hematology test, then be warned, because the problem is that myeloid cells lyse fairly quickly. So you're going to get a skewed result. So say something, for instance, like a JAK V617F. After 3 days, the number of myeloid cells that are still around that you can extract DNA from is way lower than it was before. So you could end up getting a skewed result and a false negative. So sample integrity counts, and if it's not in a PAX gene tube, then the time it's not protected. So ICE protects it, and EDTA is obviously critical, but the time it takes to get to the lab as well. Sorry, I'm speeding up a bit. Okay, so make sure, this is now where you as a clinician, where it's important is make sure that the mutation you're looking for was actually assayed. And that seems a bit silly to ask that, but it can happen. So say, for instance, you have a FLT3 inhibitor that you possibly can use if you're lucky enough in private, then you ask for FLT3 mutations. Now, the result might come back saying FLT3 mutations not detected, but you need to make sure that the lab actually looked for internal tandem duplications and tyrosine kinase domain mutations. both are relevant here. So make sure that you read the methodology, and if you don't have the methodology on your report, then ask again. A lot of labs just assume that FLT3 mutations are only the ITDs. So you could get this, and I've seen it in a report where they really actually only looked at ITD. So this again could have clinical implications. Make sure that you understand the significance of the results. So, you know, this now is cat and Hansa binding protein alpha report now says it's detected. Well, this actually only has, this mutation actually only has clinical significance if it's a double mutation. So if the lab doesn't report that, then that's not very clinically helpful. And whatever you do, don't get misled by qualitative assays trying to give some kind of quantification result. So anything that has peak heights, peak areas, band densities is absolutely meaningless. These are relative to the assay that was run at the time. They are not, they are highly dependent upon when you run the assay, your reagents. So you cannot do a serial assessment for this, for instance, of a FLT3 and just look at the peak height of the mutation over time. That is not at all how these assays work. So it is purely a yes/no answer. So in terms of qualitative assay examples in South Africa, I changed this now to the NHLS because I'm sure Theo does a lot more. I'm very jealous. You'll see that I've knocked off the list FLT3 ITDs because now that is no longer a yes/no answer. That is now a, we use the allelic ratio, which means now it's moved into the quantification kind of range type of assay. But the others here you can see are your typical NPM1s, your JAKs, and your NPLs, for instance. The things like T-cell clonality and your B-cell receptor clonality, these are particularly problematic when it comes to using, for instance, a lot of labs were using the in vivo scribe kits. These are incredibly large genes, and you actually need many PCR reactions to be able to look at the entire region. So what happens is that's an incredibly expensive exercise. So not all labs look at the whole area. And I've definitely seen that where a couple of labs will just choose spots that are commonly mutated. So the problem is then that when you get a result that says it's negative, it basically means that the patient hasn't got one of the common kind of monoclonal kind of mutations. It definitely doesn't mean that there isn't a monoclonal population. So this kind of thing is really important to be aware of. Okay, so now when it comes to quantitative assay interpretation, this is usually RNA based, thus is time sensitive. RNA degrades very fast. And unfortunately, it doesn't degrade, different transcripts don't degrade at the same rate. So there might be different, some are incredibly stable, but a lot of them are not. So you need to take some kind of precaution to be able to protect your sample, whether that is a PAX gene tube or whether that is time and refrigeration. Those kind of things will help limit the degradation, but it's not foolproof. And unfortunately, you can have problems here. So while I'm saying it is usually RNA based, I did just mention the FLT3 ITD is now regarded as a quantitative assay because you're looking at the allelic ratio, and that's now relative quantification of the basically the area amplification of the mutated peak over in the same tube, the amplification of the wild type. So this is relative quantification. And STR chimerism via STR PCR genotyping, they both use capillary electrophoresis and they have a 5% sensitivity. So they're not very sensitive. And something like the allelic ratio, for instance, I'm not quite sure how NGS works for this, but the ELN at the moment with their guidelines, their cutoff of 0.5 ratio, that is very much defined as using a capillary electrophoresis peak area only. I'm not quite sure how the NGS, when you are trying to get the allelic ratio, how that then compares, but you obviously also have to be aware of that. Really huge, and I think this is the biggest thing, a take-home message. If it's anything from my talk, it's this one, is that gene expression, when you're looking for in the quantitative assay, is always assessed as a ratio or a percentage. Copy numbers on their own are not relevant. It's only the ratio or percentage. And this is, you need to be able to express your transcriptive interest against a housekeeping. And this is because there are so many steps in a quantitative assay that can change, and lots of different reagents that can change how well something amplifies, literally tube to tube. So just because something has 5 copies, I can, and we've seen it with our BCR-ABL quant all the time. I mean, you can have 200 copies in one replicate and 1,000 copies in the other. But when you're amplifying and you compare it to the housekeeping, the number of housekeeping transcripts, then they balance each other out and the ratios are exactly the same. So this is really, really, really critical. Quantification assays are very specific. So make sure that you're actually asking for the correct thing. So for instance, if you ask for a P210 quant, if you just ask for a BCR-ABL quant, I'm probably going to come after you. But if you ask for a P210 quant, we're not going to be able to detect P190 transcripts from that, using very specific primers. There is a chance of false negatives, and there is a low chance of low-level false positives as well. So if you are using the wrong primer set. And the sensitivity depends upon the techniques and the transcripts that are being assessed. So not all disease assays are able to go over 5 logs because it depends on how much transcript is being expressed. So for instance, BCR-ABL to ABL in a cell is 1-to-1 in an affected cell. PML-RARA to ABL in an affected cell is 1-to-10. So it's already a log. The amount of disease you can detect is already a log less. It's simply because of the amount of transcript in each cell. And as I've said here, that quantification assays have different sensitivities, even for the same transcript, depending on what assay you're using. So you need to know what the assay sensitivity limit is of a test to be able to interpret it as a negative result. I think one of the key things here is definitely, please don't do this. Do not change to a different lab or monitoring using a different assay while you're monitoring for minimal residual disease or from diagnosis to minimal residual disease. These ratios, these kits or different labs could be using different assays. The housekeeping genes are different, and the ratio that you're going to get of the transcript of interest to the housekeeping gene is not going to be the same, because it depends on how much of that housekeeping gene is actually expressed. So they could be very different. So the ratio that you get from one lab using one kit is not the same as the ratio generated using a different kit from a different lab. So they're not comparable, and I'll show you examples of that now. So here's a couple of examples of kind of what can go wrong when you, especially with the quantification assays. So this was a case of an APL patient before and after consolidation therapy, where you actually really don't want to see any transcript with an APL patient. But clinicians decided all was well because there'd been a 2-log decrease because they were purely looking at the copies. So this, for instance, was report A, where they saw 23,500 copies, and report B had dropped down to 425 copies. So they were like, wow, great, 2-log decrease. But the problem is, you can see here that the actual copy number of the housekeeping gene, which in this case was actually FD8, which is a horrible housekeeping gene, varied radically. So when you actually, and this could have been the sample was degraded, the amount of maybe the reverse transcription wasn't very good, so the yield was a lot less. Nothing wrong with the actual assay, but the copy numbers are then lower. So when you actually look at the ratio, which is what you want to look at, then you'll actually see that the ratio for both reports is exactly the same. So in other words, there was actually no response to therapy at all. And here I just wanted to point out that if you times this by 100, which is now going to give you 7%, that does not necessarily translate to 7% disease. I think we've all been conned a bit with the BCR-ABL IS percentage. You know, you just times by 100 and that gives, well, that's great for BCR-ABL, but it doesn't work for something like PML-RARA, for instance. That only works in a case where the transcript and the housekeeping gene are a 1-to-1, which is very unusual. So please don't ever interpret that ratio other than BCR-ABL as like a 7% for instance. So this is really dangerous because the clinician had decided that the patient was doing fine, and yet there was no response at all. So that was not good. Here's another example where if you ask for the wrong test, you can actually get a false negative. So this was a patient that was diagnosed many years ago using FISH, 922 positive. They were put on imatinib, which CML makes sense to me. And they were monitored by quantification P210 assay for about 10 years. And they were always negative. And they were always FISH negative. Great, no problem. But they were not monitored at diagnosis for the P210 assay. After 10 years, strangely out of the blue, developed weight loss, poor appetite, didn't make any sense. So a bone marrow was performed and actually showed evidence of accelerated phase CML. You have to ask yourself, how is that possible? Well, we actually did a multiplex BCR-ABL on that patient and actually showed that the patient was and had probably always been a P190 BCR-ABL. Now that's incredibly rare in a CML, but the point is it happens. And if you don't confirm something like a FISH or a karyotype, a 922 karyotype at diagnosis, even in something like CML, if you don't see a positive result within 3 months of therapy, then you can pretty well put money on that they were never a P210 transcript. We also showed that even though imatinib would have worked against the P190, but actually what had happened was that this patient had actually developed a mutation in their P190 transcript, in the protein, sorry, of the P190, and they'd actually developed an imatinib-resistant mutation. So they needed to be changed to a different TKI, and then fortunately they've managed to kind of salvage that patient. But it's really possible how things can mislead you. The other thing was that this particular lab, unfortunately, were lacking some expertise and got caught up in the false advertising of the kind of machinery in the kits, and this was with the gene expert. So this was a CML query patient, and a P210 quant was requested. And the report came back saying that it had been detected and the IS percentage was 0.003%. Couple of issues with that, but before I get to that, they then came to us at the same time for a JAK mutation, and it was positive. And obviously the clinician was confused. I would be. Now, the thing is that this was a diagnostic sample, so it's highly unlikely that the patient is going to be positive for JAK and for BCR-ABL translocation at the same time at diagnosis. After you've been on therapy for a while, you can develop another clone. It's usually, well, it can be either way around, but it's highly unlikely at diagnosis in untreated patients that you're going to get positivity in both. But this is the kicker here, is that at diagnosis, you are not going to get a CML patient with that low level of BCR-ABL. That's just not going to happen. And the biggest problem is that no current assay, and I'm going to say current assay, including the Gene Expert Ultra, can actually detect, I'm going to put in inverted commas here, a true transcript below 5 logs. So anything below 0.001% is more than likely going to be a false positive. And it should have been reported as negative. So if you get a report below 0.001%, please take that with a cautionary note if that is positive. Because even this assay cannot. It can give you a false positive result, but it's not necessarily a true positive result. So the kind of take-home message from this is, know something about the assays you're requesting. And in this case, that most quant assays report in the 4 to 5 log range. And know that obviously at diagnosis, this is a nonsensical result. So this unfortunately was one that I got wrong. So I'll take some of the blame for this one. But this is where not giving the lab any information can also lead to a false positive. So this was a CML patient who was 87% fish at diagnosis. I did not know that. They were P210 negative at diagnosis using the gene expert. I did not know that. And they asked us to do a P190 quant, as I said, with no information supplied, whether it was diagnostic, whether they were treated or not. And we were asked to do the P190 quant after 3 months of therapy. I did not know that either. Okay, so we came back with, we had detected P190 transcripts, and it was in the 0.01% range. So our report, unfortunately, said that the patient has achieved the equivalent of a major molecular response, and the transcript will not be detected via fish. Okie dokie, sounds fine. However, it was very hot for fish. So before I kind of pulling my hair out and thinking, oh my goodness, what is going on here? The key thing is that this is where it's really important when you do MRD that you are, and you're doing quantification assays, that the input, that the test is the correct one for the job. When it comes to BCR-ABL, there are several different isoforms, and unfortunately, the primers can actually bind to different transcripts, but will give low-level amplification, but will give high-level amplification for the correct one. So what we did was, when this result came back, to try and troubleshoot what was going on, we put the sample through a multiplex assay, and there, lo and behold, lo and behold, it actually was a very hot P230 BCR-ABL positive. Again, strange CML. So what had happened is that the P190 primers can actually bind to the P230 and give you a low level. If we'd known that this had been a diagnostic sample, then I would, number one, known that this was totally didn't make any sense, and put it through. You want an assay that can pick up the different isoforms, especially with it being P210 negative. So yes, so unfortunately, we can get it wrong no matter how good our assays are, no matter how many controls we run. There is this element of false reporting if we're not given the correct information. This was it was an interesting case where this was a kiddie, was an AML with inversion 16 and was KIT positive at diagnosis. Not sure the KIT positivity actually has much influence over the inversion 16, but 1 month after, so post-induction, they had sent the sample for inversion 16 real-time PCR for minimal residual disease, which was absolutely perfect thing to do. Not quite sure what you would have gained from doing the c-kit. It can't be used for monitoring. The important thing here is your inversion 16 transcripts. They are what actually are causing the disease. So that is what you want to monitor in a case like this. This just aids with prognosis. But when I was told to help, all I was given was information. Clinician said, oh, it's 4.8 copies. Already I'm a bit nervous about what I'm going to see. 2 months later, they'd sent her for PCR positive, PCR again, and now came back with 198 copies. So the clinician now was upset. And after 5 months, they viewed it as the patient is still positive for PCR. So they were worried that the patient hadn't achieved molecular remission, whatever that actually is. They were struggling to do c-Kit monitoring, and they were wondering whether or not the patient should actually go to transplant. So one of the key things, so I really wouldn't recommend trying to do c-Kit monitoring in a case like this. Purely that mutation is only for prognostication. The big problem here was that this test was done at a different lab to these tests. So different housekeeping, different lab, different housekeeping gene. Forget the fact that the copy numbers are irrelevant, but actually you couldn't even use the ratios, would not have helped you comparing this result to this result, which makes that just a nightmare, literally nonsense to try and figure out. So taking a closer look, so the first result couldn't actually be used to compare at all, because it was actually was using ABL as a housekeeping gene. So the ratio, there's a lot less ABL, so the ratios are much higher. So it really doesn't help you at all. So just looking at the two that were from the same lab, this was good report in terms of had the copy number and the copy numbers of both the gene and the housekeeping gene. I wouldn't actually recommend putting those in because, as you can see, it confused the clinician, but the ratio was given. And in this case, it was 0.003, the ratio. And then after 3 months, it went down to 0.508. So this had been a massive reduction, almost a log reduction, which means that it was heading in the right direction. So unfortunately, couldn't compare it to the 1 month before because it was the wrong, the ratios would have meant nothing. There was no diagnostic quantification done, which is really essential in the case of an AML, because you want to look for a large, like 4 log reduction. And the clinician was completely confused by this copy number, which was not at all relevant. And also this trying to achieve PCR negativity, what is that anyway? When it comes to AML, you're looking for, as I say, a 4 log reduction or at least a stable low level. So you must be aware of what it is you're trying to achieve. And unfortunately, as the assays are getting more and more sensitive, you're going to find that more and more AMLs will still have positivity. So don't make that your end goal unless you're sure that that's what you want. So finally, I've just got 3 slides because I really don't know what I'm talking about with the NGS. So I don't, this is basically just on kind of what I've experienced, but I'm definitely not the expert on this. Currently, most of the NGS panels available in South Africa, although Thea might tell me differently, are qualitative, and I'm specifically talking about the Oncomine Myeloid Research Assay. So really it's going to give you yes/no answers. It's not suitable for monitoring currently, this current assay. There are ones that are being developed, and I know they're improving that. And that's mainly because of the noise that you get off these assays. And it can't be used for MRD because the sensitivity is normally in the range of about 1 to 2%, which is not sensitive enough. Minimal residual disease, basically, you want to be able to get down a couple of logs reduction. You do, however, get highly targeted detection of these B and T cell rearrangements by NGS, and those are quantitative. So when you start homing in on specific genes, then you can use NGS for MRD. So what's really important is that you need to understand what you're asking for when you say NGS. You need to understand what panel you're asking for. Some genes, especially in this Oncomine panel, some genes are only sequenced in hotspots. They're not looking at the whole gene. I stand corrected, but I think this is the case with JAK mutations. Some genes are sequenced entirely, which is fantastic. Others are not. Some genes have reduced mutation detection rates because they're very GC rich. So while one gene you might find a mutation that's sitting at 1% disease, whereas another might be 20%. And that's just because the technology doesn't allow it to get over those GC rich regions, which we can do in test specific PCRs. You can have an indication of allele frequency, so because it's based on the number of mutated sequences you get, so you you can tell homozygous, for instance, for some mutations, as we can in other assays. But the key thing here is that you can't look at chromosome copy number in this Oncomine panel. So you really need to know the panels, and sample quality is still an issue. You're not escaping from that, whether it's RNA or DNA input. In fact, I think it's more of an issue with NGS. So in South Africa currently, the commercial kits are using defined pipelines. So that means basically the whole system is defined as to how to interpret the data. Predetermined reference sequences, as far as I know, and you get the bioinformatics system is set up so that the NGS data, so these NGS reads that you get, are compared to a reference sequence. And you basically, it's going to call a variant if it passes all of the internal QC data from this particular pipeline. So, and then what happens is if a variant is called, like in this case, a SNP, so there's a mutation now that's been seen on more than one occasion, it gets screened against databases where clinical variants have been reported, and then so basically it's an extended literature search of sorts, and whether or not these mutations have been linked to anything of clinical significance, and then these are viewed as tier 2 variants. So I'm not going to pretend that I 100% kind of, yeah, no, I'm not going to pretend I'm an expert on this, but this is the minimum that you should get in an NGS report. You obviously the gene of interest, the amino acid and nucleotide change, the allele frequency, so that's very important. So if it's more than 50%, that would mean that it's got to be homozygous in some cells at least. And then whether or not there is this, whether or not there is any clinical significance. If you are lucky, so this is the minimum, if you are lucky, and I would highly recommend you pick labs that have given some kind of interpretation for this. It's really important to kind of put this in perspective. So for instance, in this case, as an example, is that 31.22% basically means that this is probably a single mutation. It's not a double mutation at the site. And then that actually doesn't have much clinical relevance in a cytolink normal AML risk stratification setting. So it's really nice if this is kind of, so just because you see loss of function here, don't throw out all the other literature that says that, in this case, that a double mutation is what's important. Or if it's only a single mutation in this gene, that this has got to be more than, it's got to be homozygous for that mutation. I think the worst that any clinician or pathologist would want to see is that there's no variants detected, no fusions detected, because now detected, what does this mean? And I can tell you what it doesn't mean. So what it doesn't mean, like in the Oncomine panel, for instance, it doesn't mean that there's no copy number variations. It doesn't mean that there are no chromosomal rearrangements, because there are specific ones that are looked at. This is not a karyotype, so it does not look at everything. It doesn't mean that there are no point mutations anywhere, because the panels are gene specific or even hotspot specific. And you can also have issues with FLT3 ITDs. I know that they've improved things a lot, but depending on where they are and how big they are, you may get a false negative for that. And there are issues with the CAT enhancer binding protein. Alpha can also give false negatives because of the GC-rich region. So you need to bear those all in mind when you get a negative report. And my last slide is basically, after all that, and even I'm tired, what to test, when to test, where to test. Ask your hopefully friendly molecular person. We are here, this is what we do, this is what we concentrate on. We try to keep up with the literature, we try to keep up with how to interpret things, the new technology, what is clinically relevant, what is not. Ask us. There's no way we can be expecting this day and age with things moving so fast for clinicians and pathologists to be able to keep up with this. So please ask us, and there's lots of us out there. I'm not just talking about me. There's lots of us, including Theo. So, but the bottom line is rubbish in, rubbish out. So know what you want to see, and ask those specific questions. And that's me.
Thank you very much, Karen. That was a really brilliant, very practical overview, especially of the PCR tests. I'm not sure if anybody's actually got questions now for Karen. Otherwise, I think we'll press on with Theo, because there may be some questions that arise at the end of Theo's talk. So I'd just like to introduce Theo.
Jessica, sorry, just before you go on, I see there is one question in the chat box.
Oh, sorry, I've just seen that now.
Okay. Yeah.
Okay, Karen, I'm not sure if you can see the question. It says, are the BCR-able qPCR tests the same from lab to lab, especially the P210 assay?
That's pretty much the only one. No, they're not the same, but because it's really the only one that is standardized. So because we report on the BCR-able international scale, then results are actually comparable between labs. It's not perfect. It's definitely not perfect, but it definitely is the only one that you can theoretically jump from one to another, because it's on this. We've kind of normalized each other to a much, an international number, but that is the only one. None of the others are like that at all. None of them. So yeah.
Thank you, Karen. And then there's a comment from Pascal Willem. Good afternoon, Pascal. Theo will probably comment, but there are a lot of pluses in NGS. Yes, especially regarding the number of aberrations detected in a single test. Yes, absolutely.
Yeah, no, so, I'm not. Yeah.
Yeah. So I'm going to move on to.
I'm not an NGS expert. Yeah, no.
So I'm going to move on to introducing Theo, because we've still got a lot of exciting material to cover. So Theo is actually started off as a hematology pathologist trained at UCT, and then went on to specialize in clinical hematology, and he qualified 10 years ago now. And he is now working in Pretoria at the Albert Stem Cell Transplant Center, and he's the medical director also of OncoLab Diagnostics, which is affiliated with the Stem Cell Lab, and has a lot of experience in setting up molecular assays, genetics, FISH, quality assurance, and next generation sequencing. So I think it's very interesting to hear from Theo the perspectives from the private sector in this regard. So Theo, over to you.
Thank you very much, Jessica, and thank you, Karen. I'm just going to try and share my screen quickly. So can you all see that?
Yes, I can see that.
Okay. So I would like to briefly take you through my experience after having trained and worked in the NHLS and then going to private practice. It was quite a culture shock, and it felt to me as though I had taken a couple of steps backward rather than forward. And I would like to explain to you the pitfalls of testing in private practice in South Africa. So some of the issues Karen mentioned are doubly worse. So, just some disclosures that I did consulting for. Okay, so I'm going to talk about karyotyping, cytogenetics, FISH, PCR only a little bit, because I know far less about that than Karen. Then next-gen sequencing, where I'm taking a lot of my colleague Robin Marshall's work and reporting on that. And then MRD, which is one of my passions. So private pathology in South Africa is extremely competitive, as you know, and often the politics, unfortunately, interfere with patient management. Our biggest problem, and that the NHLS is far ahead of us, is that we have no central data repository. If a patient comes to you having had his diagnosis elsewhere, the chances are a lot of the diagnostic tests will be repeated because you cannot find the reports. Clinicians can be very adamant, and the balance of power in private practice is so that pathologists say yes to everything. They do not reason, they do not argue, they do not question, they just do the tests. Reimbursement considerations are a problem, and often I've had to write motivation letters for tests we do that weekly, and unfortunately also there is an element of greed. Reports are not the same as they are in the NHLS, sometimes grossly inadequate, although some labs make very nice consolidated reports available for their patients. The onus of interpretation is often shifted from the provider, from the pathologist to the client. And this is not laziness necessarily on the part of the pathologist, but often because they have very limited data, or the test was grossly inappropriately requested. So nobody knows what to make of the answer. And there isn't this relationship between the pathologists and the clinicians where they can discuss backwards and forwards whether something is appropriate or what is going on. So those are all things that hamper us. And I would like to put a disclaimer in here that many of the things I mention now happen in my own lab. I'm as guilty of this. So the first example, a patient with erythrocytosis, JAK V617F mutation, was tested 3 times in the space of 7 months. This with the same lab. The lab will not query it. They will test again and again and again as long as you send them samples. All of them were negative. Example 2, flow cytometrical MRD in brackets testing performed at day 14 of induction of AML. Example 3, a full FISH panel for a myeloma patient performed on a sample with fewer than 1% clonal plasma cells with no purification. Example 4, flow cytometrical staging of a Hodgkin lymphoma. Now, the only lab that I know that can reproducibly do this is somewhere in Spain. Nobody else has been able to do that worldwide. Example 5, FISH performed on a diffuse large B-cell lymphoma where the bone marrow wasn't involved. Example 6, which I see on a weekly basis. I get phone calls from oncologists and other clinicians on these patients with TALL. We do the flow, it's MRD negative, and they send it for a qualitative T-cell receptor assay, and it comes back as clonal. And they want to know, how can it be clonal if my flow is negative? And I still haven't brought that home to them, that after treatment, after therapy, one gets these oligoclonal bands that can mimic clonality, and they do not represent disease. Also, what we found out with one of the big private pathologists is that they do not compare on the fragment analysis, the clonal fragment, they do not compare it to the diagnostic one. So it might be a completely different T-cell receptor that's now coming up clonal. So I wish they'd ban these tests because it just confuses everyone. And then example 7, we've had a couple of IGHV tests asking to be repeated, or where the patient was in remission and we couldn't find any clonal population. All right, so karyotyping and cytogenetics, you are very lucky in the state service. It's not readily available in the private sector because it's low labor intensive and low income. The pathologists are not very interested in it. We on average have a turnaround time of about 6 weeks, even for them just to report to me that they didn't find any metaphases. So that's very frustrating. Also, it seems to be that the pathologists do not request it on new leuks. I've had to several times remarrow a new leukemia patient just to get the sample for cytogenetics. This is definitely not everyone, but it happens. Chronic lymphocytic leukemia, where we should be looking at the karyotypes. We don't do this at the moment, but those who do send samples in, please make sure that your lab is stimulating their metaphases. Either with TPA or CPG is the preferred one at the moment, and could get your metaphases in up to 80% or more of patients. Then there are some alternatives for karyotyping in FISH. The Affymetrix microarray is in Pascal Willem's lab. I know very little of this, but I do know that they use it for copy number variation. There is in private practice an instrument that does comparative genomic hybridization. Now, just in layman's terms, both the microarray and the comparative genomic hybridization is like a method where one measures patient sample, be that DNA, RNA, or what, you measure it against an equal amount of a control sample or a normal. And then the patient sample is labeled in red, the other sample is labeled in green, and then if you get for each little spot, you get more green or more red, you know you've got more or less compared to your control that you put in. The comparative genomic hybridization instrument that is placed in Pretoria in a private lab, unfortunately, I think is only used for constitutional abnormalities. It is very difficult to detect low level, like a fish with only 10% of cells where 7q is deleted. That will not be very easy. But if you have 50% deletion or 100%, whatever, that's much easier to see. Next-gen sequencing, we tried out copy number variation, but it has similar problems to the prior methods mentioned. I'll show you quickly. It can detect translocations. And then there's this thing I got very excited about, the BioNano Sapphire. And I think 2 or 3 of these instruments have been placed in South Africa. I don't know where. The BioNano Sapphire is, and the stocks went through the roof in the United States of this thing. It's like a mixture of a hybridization experiment and sequencing. It can do whole genome copy number variation. It can do whole translocations, including novel translocations, and it can do complex karyotypes. About 4 days. So this is the instrument, and it's a chip-based thing. So basically, this is what they claim to be able to detect, and they have got very robust data. The problem with this is one needs to isolate high molecular weight DNA. When I say high molecular weight DNA, this is in the region of 100 kilobases and larger into the megabase scale. So one's normal DNA isolation procedures do not work for this. They have a special kit which one needs to work. Your tissue needs to be either alive or snap frozen when one works for it. So when one uses it, so formalin fixed paraffin embedded tissue is useless. So I very much wanted this instrument, but it isn't the be all and end all. It's not going to replace fish and cytogenetics in a lab, which is what one would want at the end of the day. Also, the other detractor here is that in the United States, they have a very competitive pricing schedule for these chips that one pays for a whole genome. One pays about between $450 and $550, and you have to do no karyotyping, no fish on that. They do not have the same pricing structure in South Africa. It's probably going to be closer to $1,000 for the chip, and then it does not include the instrument, which is included in the United States. So we are fighting with them still to get a better pricing structure for third world countries, but so far nothing. Fish, just quickly, it's readily available. The turnaround times are generally fast. It's labor intensive, although some automation with karyotyping systems may be used. In the state service, reimbursement is definitely a problem, and I think I've written the most letters about this assay. Plasma cell myeloma, please ask your laboratory, look at your aspirate, see how many plasma cells were counted, and ask them, did they purify or sort these cells before doing the fish? If you have 10% plasma cells, your sensitivity to pick up a TP53 mutation drops significantly. And the same for chronic lymphocytic leukemia. If you have 70% or less clonal B cells, Eric, currently, the European Research Initiative on Chronic Lymphocytic Leukemia currently recommends that one purifies these cells before testing for TP53 via with molecular methods or FISH. So please ask your lab and see if they actually purified your cells, because otherwise you are wasting money. Okay, the polymerase chain reaction, or PCR, I'm not going to go into a lot of detail. A couple of labs do chimerism. There's different technologies, the STR and the qPCR assay. They're not always comparable, but generally they are. Most of the studies looking at their clinical value have been performed on individual cell fractions like blasts, T lymphocytes, and specific disease scenarios like childhood ALL also. The timing of testing after the transplant is important, but not specified. So the most important takeaway from chimerism chimerism. And I see this in my own practice, where I see patients post-transplant, and I have a 90% T lymphocyte chimerism at week 6 post-transplant. What does one do with this? So one needs to develop clinical algorithms that say, if I have my granulocyte or my T lymphocyte or my whole blood chimerism at this period in time is less than so and so, I am going to give a donor lymphocyte infusion, or I'm going to give a stem cell boost, albeit what you want to do. And then one needs to go and retrospectively evaluate whether your actions were correct. Because if I publish a chimerism report and the clinician phones me and says, what does this mean? There's very little literature available to tell them what it means for the patient. We know that if it suddenly drops or so, and it's a dramatic shift, then probably there's more significant, but there aren't clearly defined cutoffs, and everybody uses their own. So BCR-able, most private labs will use the Gene Expert P210 assay, which will only detect the E13A2 and E14A2 transcripts. So we actually issue a disclaimer on this. One needs to know the assay sensitivity and limitation and the individual sensitivity of a patient sample. Definitely you need to know the difference between the P190 and P210, and please demand a proper report with at least the minimum information you need. So I'm just going to show a report here we have of a patient that was recently tested, and one can see, yes, they call it positive. They report the old transcript still, the nomenclature is wrong, and then they say this developed or detected at a ratio of 13%, previous one 86, previous one 76. They don't report sensitivities or copy numbers or anything like that. So we based our reports on those of Wits and Karen's lab, and so this is one of our reports, and we will report the BCR-able, the international standard ratio, and also the copy numbers, even though I hear Karen said that may be confusing. I think it is less confusing so if one gives a visual representation. And as Karen mentioned, the Gene Expert has been validated up to a certain level of sensitivity, and that's MR 4.5. So previously, we drew this red line, and if it was below that, we reported it, and we said we are unsure of the clinical significance thereof. However, the Gene Expert has released a new version of their kit, and they have actually, they will no longer report. So the instrument will call it negative if it goes below this, and they don't give you that information. So they've obviously realized that there was a problem there too. Okay, next generation sequencing. So here HLA typing is the big winner of the NGS era. It is inexpensive and relatively fast. So when I say inexpensive, it is probably less expensive than doing serological typing followed by intermediate to high resolution typing. And it gives you high resolution typing from the word go with some caveats. It's relatively fast. So our workflow at the moment is very laborious. So it takes us about 5 days, more likely 7. Data interpretation can be a bit of a bottleneck, and it's still in its infancy. So the bioinformatics changes quite frequently. When we started with HLA typing, I thought this was going to be the last word. In HLA typing, we would be able to report everything, but it's not true. So it's often unable to resolve ambiguities in especially some class 2 alleles in DQB1 and DPB1. It's because the exons and introns are not fully covered by the current assays and probably won't be by future assays. And this is independent of platform. Illumina will come to you and say you can only do HLA typing on their instrument, and Thermo Fisher will do the same. I can tell you that we have both instruments in our lab. The problems persist. It's not because of the technology, it's the assays. So there are many repetitive sequences which create alignment errors leading to ambiguities, and then changes in the 5' untranslated region 3' UTR may change expression of these alleles. So the stem cell registries, actually recommend that we report the ambiguities as they are, or use the G codes where these ambiguities are grouped together in specific genetic codes, but you shouldn't go and do Sanger sequencing to resolve all of them up front. That can be done at a later stage if needed. So just the HLA, this is a report we would issue. So if I can just go to this HLA-A allele. So the first field, this 31, would be the antigen, which in the past was serologically determined. The second field is the allele, which is determined through the DNA sequence. The third field is determined by synonymous nucleotide changes in the exon. So this doesn't actually change the sequence. And then there's a fourth field, which we don't report because it massively increases the number of ambiguities. And the fourth field relates to changes in the non-coding region, so the untranslated region and the introns. And then there are suffixes like N, which means that antigen is not expressed, or S, which means it's only secreted. So we don't report all of that, but we try and resolve up to the third field. As you can see with this HLA-DQB1, that wasn't possible. And so at the bottom of this report, we list those ambiguities. It is safe to accept that if you are testing a sibling or a related donor, you will get the same number of ambiguities, and it won't be necessary if you choose that donor to specifically go and resolve them. All right. Then myeloid malignancies, which is probably our biggest use of our NGS instrumentation. I recently went through our data and we've sequenced about 370 patients in the last couple of years. It's indispensable for prognostication and monitoring of clonal myeloid neoplasms. Here we look for nucleotide and amino acid changes. We don't look for big structural changes or anything like that. So point mutations and short nucleotide, polymorphisms. The data analysis, despite everybody claiming that it comes with bioinformatics and this and that, is very time consuming, especially for myeloid variants, because they are not properly annotated. If we get samples for solid tumor analysis, those take you 5 minutes to sort out, because there's thousands of solid tumors perfectly annotated. Just because our diseases are so much more rare, these haven't been annotated. It's compared to a reference database, so HG19, human genome 19, or human genome 38. And these databases have some idiosyncrasies. So there will be a SNP in this database, and then 90% of the general population will be reported as having a variant at that sequence, whereas it was actually the reference database that had the variant. So we filter this data, and I'll show you now, and then we have several databases of annotated variants. Often they contradict one another, but Cosmic, ClinVar, DBSNP, UCSC, Varsome, IARC, and the rest. And then there are computational databases that can go and use computational models to say whether a variant that isn't described elsewhere is likely to be pathogenic or not. One needs to know the panel used. So most of the labs use this Oncomine Myeloid Research Panel, but there are some of the big pathologists who have developed their in-house panels. I would strongly advise against this, because we get to the next point, blacklisted variants. We know that this Oncomine Myeloid Research Panel cannot detect certain variants in CEBPA, certain very large FLT ITDs, and one mutation in ASXL1, which is also in a GC-rich region. If you develop an in-house assay, you will not know this. And during our correlation studies, we did find that because their primers were in different places, there wasn't a very good correlation between results, whereas when we correlated with another lab doing the Oncomine Myeloid research, we had virtually 100% correlation. So one needs to be very careful before you dive in and start doing your own thing. Although with the proper knowledge, it's perfectly possible. It's just you will never be able to fully validate your assay because you won't have the different mutations available to test. Okay, one needs to ask about read length and fusion detection. So fusion detection is usually with RNA input to detect gene fusion. So this is available on the Oncomine Myeloid Research, but not all other assays. The read length, so for FLIT and for many of our others, we've had to increase the read length. So the fragments that you analyze from 200 base pairs, specifically this assay goes to 400 base pairs to pick up on those FLITs and some of the other things. This myeloid assay stretches the limitation of most NGS platforms due to the highly repetitive sequences, perhaps not PacBio. PacBio is a platform that is not widely available in South Africa. It uses a specific technology that goes for far longer read lengths of in the kilobase, up to 80 or 100 kilobase reads, but not widely available in South Africa. Okay, so when one finishes a sequencing assay, as for the myeloid, for instance, you end up with between 20 and 80 million sequences. And these sequences are all between 200 to 400 base pairs in length. So they need to be aligned to the reference sequence. And this is what takes hours of bioinformatics, where we leave the instrument on overnight to do. And you end up with something like this. And you can see this is a chromosome P13.1. I think this is chromosome 1, but I'm not sure. And you have all these sequences that aligned, and you'll see at the bottom here, some of the sequences are cut off, and that's where they ended. So one tries to find the best fit. There are also these little spaces where a sequence wouldn't align, and then this shows you some nucleotide changes in the sequence as well. Once you have this data aligned, we go and put it through filters, and this is where the bioinformatics comes in. So when I put through the data unfiltered, this is one patient. If you look at the bottom right-hand corner here, you'll see that it found 2,421 variants. Obviously, this patient doesn't have that much, and one has to go and filter it out further. So we use a cutoff of 5% or more to call anything on our myeloid assay, because we feel the noise below that starts to get statistically problematic. And then there are quality scores and lots of other things that we use to determine whether a variant is true or not. The supplier, Thermo Fisher, also has a standard filter, but we found that filter to be overly restrictive, as it will only call variants that have been annotated and documented in COSMIC, the database. So you'll miss things. Okay, and then if I look at the same patient, the fusions, it picked up a range of fusions, 721 in total. And obviously many of these were not true. So then we put it through our own in-house filter, and we end up with 15 items. And then we have 3 people, so Robin, I, and another one of our scientists will manually go and put each of these variants through all the databases and check, and we will make a call individually, and then we have a combined meeting to discuss, like a molecular board meeting once a week, to discuss and see whether we agree before we issue the results. And then obviously we have this concept of a VUS, a variant of uncertain significance, which hopefully as we go along will become less and less frequent an occurrence. That's where none of the databases have reported the variant, or where they truly don't know whether this is significant or not. Okay, and then this is our report, well, a summary of our report that goes out in the end. So we mention most of the things Karen mentioned, and also the breakpoint regions for our fusions. And you can see here at the bottom, we mentioned the methodology, but we also have this disclaimer. Variants not detected by this assay include ASXL1, the CEBPA, and those and FLT3 IDH mutations more than 176 base pairs. What some labs overseas, we don't do it at the moment, do is that they, for these specific blacklisted genes, they will go and do a PCR with capillary electrophoresis to detect these. And certainly for FLT3 ITDs, that is preferable, because an NGS turnaround time of 2 weeks for a FLT3 ITD when you have a FLT3 inhibitor available is far too long. Okay, quickly through this, the TP53 we do with a specific kit. TP53 guidelines through ERIC and accreditation for CLL is available. Sorry, I need to go back and just answer Karen's question here about the allelic ratio of FLT3, which is very important. We kind of use the variant allele frequency of FLT3 as a surrogate marker, but it is not comparable and it is not as stable as an allelic ratio that is detected by Sanger sequencing or capillary electrophoresis. And there have been a couple of publications and people get very upset with one another who want to use this variant allele frequency, but it probably does not give you the same information that one wanted. Okay, so sorry, TP53, we look at the variant allele frequency here. As per ERIC guidelines, we do not report on anything less than 10%. We sort samples with less than 70% of clonal lymphocytes, and we perform reflex testing after agreement with our clinicians. If they want for a CLL TP53 FISH, we will also do the sequencing. And obviously this is repeated at each new treatment interval. IGHV, ERIC also has extensive guidelines and accreditation available. At present, we still use the Biomed primers, which have been very well validated. We are busy looking at the LEADER assay, which ERIC has made it their assay of choice, and they require that one runs the LEADER assay to be accredited at the moment. This test is only performed at diagnosis, so please do not repeat it. It's a constant parameter. We will report, on our report, we will state, is IGHV mutated or unmutated? If it is rearranged, and then if it is rearranged, so clonally rearranged, we will say, is it a productive or an unproductive rearrangement, which basically means, is this B cell receptor expressed or well, translated and expressed on the cell or not? And then we also we have a different bioinformatics tool, which we go and look for the stereotype. Please request this data if it's not reported, because it's available. If one sequences IGHV, you can determine the stereotype with that same data. One doesn't need to do anything further rather than run it through the bioinformatics. Okay. And then copy number variation can be done with next generation sequencing. It's not an ideal platform for this, especially not for the analysis of somatic samples, because of the same problems with CGH and the other things mentioned. Small variations in sample dilution might make you miss things. It is, however, well validated for constitutional abnormalities, and we tried out this ReproSeq from Iron Torrent in our lab, which is a whole genome, low coverage DNA sequencing. And after running it through the bioinformatics, one gets something like this. So this was a patient we knew with an isolated 5q minus, and you can see that it picked that up successfully. But this patient had about, I think, 60% positivity on FISH, so it was easy to pick up. Had that patient had only a 5 or a 10% positivity, as you can see at this scatter plot at the bottom, it would have gotten lost in the rest of the background noise. So we've kind of abandoned this for the time being. It certainly is possible, but it needs a lot of changes to your bioinformatics, which I'm too stupid to do myself. All right, then just quickly for MRD. The PCR, quantitative PCR and that are allele specific are not readily available in South Africa. We do have this qualitative T cell receptor and IGH PCR, which, as I've mentioned before, often confuses people. Droplet digital PCR is very attractive, but it's not available in clinical practice yet, as far as I know. I'll show you now, we evaluated an instrument by Biorad, and it is extremely attractive. And then flow cytometry, I'll get back to. So droplet digital PCR, what it is in brief is one um, it's like a regular PCR reaction, but it happens in these small oil droplets. So the targets are randomly distributed into 20,000 droplets. You do a regular PCR, not a quantitative or anything. The targets are labeled with fluorochromes. And then lastly, you run it through this droplet analyzer, which has a fluorometer on. And it gives you absolute quantification without the need for a housekeeping gene. Um, I say that with with some reservation, in that I would still include a housekeeping gene even if I were running this absolute quantification, because otherwise changes in pipetting or small errors might make significant downstream problems. But we use this instrument, it works very nicely. The other nice thing that Biorad has done is their website. They have over 2,000 wet lab prep kits. So you can go and put in your mutation that you picked up, and it will immediately tell you these and these and these kits will detect it, and you can order the kit. The kit is literally a set of primers, so it lasts forever. So one can potentially build up this whole library of mutations that you can test for fairly easily. So we did this with a solid tumor thing, and it worked very well. Afterwards, I got brave and tried to set up my own assay for NPM1 mutations, and it failed miserably. Okay, then to flow cytometry. One needs to know what one is asking for and when one asks for it. Check for the lower limit of detection and the lower limit of quantification of the assay, and for the chosen antibody combinations. You need to check for the sensitivity of the patient sample, which is dependent on on the sample age, and also hemodilution. This is from an article in Blood in 2018 that they gave guidelines for reporting of AML MRD samples by flow, and a bit different from the BCR-ABL. They say even below, so one still needs to state your limit of detection. So here it's 0.1% as determined with this assay in this lab. Even below that, they will still report if they detect it, but they will say it's negative because it's below the validated detection threshold, or they will say a sensitivity of when they do not detect. They will say, I did not detect MRD, but my assay was only sensitive to 1% because it was hemodilute also. Then in their report, they say standard parameters, you must comment on sample viability, the total cells acquired, the number of CD45 cells acquired, because there are very good guidelines to determine what your maximum sensitivity is based on how many cells you acquire. The blast progenitors, but anyway, and then also whether there's hemodilution, and very few of us measure this at the moment. And then one has a percentage MRD, you name the phenotype that you use to pick up the MRD, and you have a standardized conclusion with standardized terminology. Okay, I'm nearly out of time, so I'm going to go a bit faster. These two assays, I can tell you we are not up and running yet, sorry. So for lymphoid neoplasms, on the Oncomine, the Thermo Fisher assay, there are TCR and BCR, B-cell receptor short read assays. Here, it's a different chemistry that's used, so barcodes or molecular, barcodes or tags are attached at every single step of the amplification procedure, so that there isn't preferential amplification of the same genetic sequence the whole time. So at the time when one sequence, each of the single individual DNA strands has about 3 molecular barcodes on to distinguish them from one another, to improve the sensitivity, so that it's not just random amplification. So they claim a sensitivity of 10 to the minus 6. 10 to the minus 6 requires an input DNA amount of about 6 micrograms, which is ridiculous for most labs, but hopefully with prior sorting of the cells also, one can increase the sensitivity with less stringent input requirements. The turnaround time is a major deterrent at the time. I know the pediatric hematologists phone me for MRD reports before I've even received the samples. And so having a week or two turnaround time for this is no good. But we are looking at an ion torrent genexus system, which is more of it's a larger NGS system, and one can drop in samples to a certain extent. So the workflow is, one doesn't need to batch as much. And then lastly, the myeloid neoplasms. There's an assay that Thermo Fisher is developing, and unfortunately I can't show you the assay contents, but it's very similar to their myeloid research assay. They expect sensitivities of about 0.1%, more with prior sample purification. So this will detect all of the things that you detected in your initial NGS panel. Presumably you will be able to detect, but at a very low sensitivity here. It also does fusions, including PML, RARA, and BCR-ABL. I would love to see what the sensitivity of that is, but likely this won't be their primary concern because it's easier and cheaper using a dedicated assay, but watch this space. So in conclusion, in private sector, the tests required for managing your patients are mostly available, but each practice or practitioner should compile standard treatment algorithms and management of their hematology patients, and they must design the testing intervals according to their treatment and decision-making algorithm. So you need to know what you're going to do. If you ask for a test, when the answer comes back, what are you going to do? So the testing interval should be clearly defined. Once algorithms are defined, you have to engage the laboratory on whether their specific assay will answer the clinical question you are answering. Is it sensitive enough? Is it specific enough? Is it the right assay? So unfortunately, as a clinician, you are required to understand the intricacies of the available tests. You must insist on a report that is both informative and adequate for the question you are asking. And the test should only be requested based on your algorithms yet again. And then I would just like to repeat, as Karen said, rubbish in, rubbish out. Thank you.
Thank you so much, Theo. That was really interesting and really informative. There are quite a number of questions and comments, but from my side, I'd just be interested to know, you've said that cytogenetics is important. We're very lucky to have it here in the Western Cape. We've got an excellent service, and your service is much less reliable. And that the next generation sequencing, you say, is indispensable for prognostic and monitoring. What would you say is the ideal workup for an AML patient in the South African context? What order of tests, and in fact, what is the sort of ballpark cost as well that we would be looking at? Because it sounds like to me, it should be a combination of cytogenetics, NGS, and PCRs if we're going to really, fully diagnose these patients, the AML patients.
Yes, thank you. So I think cytogenetics remains the gold standard, and then some flow cytometry probably just to confirm one's diagnosis. But for prognostication, I would think the minimum would be cytogenetics. We always, if morphologically suspected, we will do FISH for APL, and then one can we usually do a BCR-ABL, even though very few patients are positive with AML. There are those de novo cases, and it does influence their management. A FLT3, if you have the drug available, then a FLT3 upfront and within a day or two would be preferable. Then with regards to NGS, we do the NGS. I find NGS far more valuable in our MDS cases, where often the FISH and cytogenetics is negative, but we do find the point mutations, and specifically the targetable ones, I would think are important if you have access to drugs. So IDH1, IDH2, some of the RAS pathway mutations and BRAF, and then what we've started to do is testing, especially in patients who fail induction. We do TP53 testing because there's some data that says that even stem cell transplant does not benefit those patients. So they are utterly poor prognosis. So at the bare minimum, I would say cytogenetics and fish for your APL if you can. I mean, if you suspect it, BCR able because you have a target available, and then if you have access to the drugs, then NGS would be nice to have. Sorry, you asked about prices as well. So the I think cytogenetics is in the region of about 5,000 rands. Karen, you'll have to tell me. I don't know what a sequence like a FLT3 with capillary like would cost. I don't know. And then NGS for that whole panel, including the fusions, would be in the, so fish probe, sorry, is in the region of about 2.5 to 3,000 rands. And then NGS, the whole panel with the RNA fusions is usually in the region of about 12,000 rands.
So that's quite a costly workup.
It is quite a costly workup. But if one takes that, the IDH2 inhibitors cost about 120,000 rands a month, then it's not.
Yeah. Okay, so we've got some specific questions here. Theo, there was a question about the sorting of samples with less than 70% clonal lymphocytes. What exactly do you mean by the sorting of samples for TP53?
Yes, so if one is going to do TP53, one does want to pick up subclonal TP53 mutations. So it is important to make sure that the sample that you extract your DNA from or that you use to test contain as many CLL samples and no contaminating neutrophils or granulocytes. So there are two ways of doing that. One can either use a magnetic bead sorting, which is available, or one can use a flow cytometric sorter to sort the cells so that you purify or enrich them. And the number of 70% was chosen not by myself, but by Eric, this standards company.
Great, thank you. And I've got a question for Karen. Two questions from Deborah Vaz. Is the gene copy relative to a housekeeping gene a relative or absolute quantitative result? And then the JAK2 real-time PCR kit uses the 1% control to calculate the quantitative allelic burden. Would this be a relative result as well? Karen, are you?
Yeah, so if you are determining copy numbers, whether you're going to make that into a ratio or not, anything that actually has a copy number, then that's regarded as absolute quantification. So anything that is compared then to, so if you're using crossing points or using copy numbers of your transcript of interest to your housekeeping gene, anything that compares something from one to each, that's referred to as relative quantification. So you can, so the gene expert, for instance, uses relative quantification, but it uses crossing points, whereas the standard beta-able methodology uses absolute quantification to get copy numbers, and then does one relative to the other to get a ratio. To answer that one. The JAK PCR uses the 1% control. I've seen quantification on that. I don't know how great that is. I've seen on some of the external QA programs that quantification is not great. I thought that 1% control was actually just a sensitivity control. But theoretically, if you are generating, yeah, no, I don't know enough about that assay. I would for the JAK mutation, the only thing that actually has clinical significance is if it's more than 50%. So if it's more than 50%, then fine, you have a number. But other than that, it actually is pretty meaningless, really. So that's my opinion.
Thank you, Karen. So we are at the moment 6 minutes over time, and I can see that quite a few people are already leaving the meeting. But I just wanted to give Pascal, Willem, an opportunity to weigh in and perhaps contribute to the discussion from the WITS laboratory point of view in the last few minutes. Pascal, you had some comments on the CLL microarrays.
Yes, sure. Thank you very much, Jessica, and thank you, Karen and Theo. I mean, that was really nice to have an overview of all the tests, and I think it coincides to the fact that we need molecular pathology. So I think we need like some teaching in molecular pathology. I think it's quite important. But regarding CLL, I think the ERIC recommendation, if I'm not mistaken, is actually related to TP53 in CLL. I just want to say that if you look at CLL above 20% of cell, you will detect copy number aberration. And in fact, the same apply for multiple myeloma. And these are quite important, especially in CLL. They've got prognostic implication. So I would really underline that, because if you look at the CGC, so the Cancer Genomic Association, you will see that there is guideline regarding multiple myeloma and CLL for copy number detection, which are quite useful.
Thank you. That's very useful. So I think I'm going to bring the meeting to an end, and I want to thank Prof. Shires and Theo for excellent presentations, very practical, very useful. I think one of the big take-home messages for those requesting tests is please to give clinical information when you are requesting the test to get the best sort of value for money in terms of your results. And I think we need to have these presentations at least once a year, because I think it's moving so quickly. And we're just delighted also to have Theo, who's just joined us at UCT as an honorary lecturer. So thank you, Theo and Karen, and thank you very much, everybody, for attending. It was a great session.
Thank you. Before everyone leaves, I just want to say from my side, thank you, Jessica, for sharing, and for our speakers. There are some great comments in the chat box, just congratulating and thanking you both for your presentations, which are really awesome. And I just want to mention next week, we will have a session on myeloproliferative neoplasms next week, which will be a joint UCT-WITS program. So look forward to having all of you here. I hope by now everybody have blocked out their Monday afternoon calendar between 4 and 5:30. It's really great. It almost feels like we're seeing each other, but not quite. Look forward to the times when we will see each other for real. But in the meantime, I can see that this is really bringing us together. So with that, have a lovely rest of the week. And there's one more question from Jacques. Where do we get the online course? Karen Shires. Karen, where do they access your online course? Which, by the way, comes highly recommended by some of my fellows who've done it in preparation for the exam, Jacques.
Okay, so it's basically you just have to email me and I can give you access to the Vula site. So it's not an official course at the moment. There are about 16 lectures, I think, and then I have short quizzes and stuff that you can go through. And I try to cover like the basics of some of the molecular techniques, and then go into how the genetics and how the testing works for specific tests. So yeah, so please just email me and I can give you access, and you can do it whenever you want.
Great. I put Karen's email address in the chat box. If anybody wants to see it and use it, I can strongly recommend it. Great. Thank you very much, everybody. Until next week. Bye-bye.
Okay. Bye.
Thank you so much. Bye.